Welcome back, everyone, to another deep dive, ready to explore the fascinating world of microscopic identification.
I'm excited. We're going to navigate the intricate pathways of bacterial identification today.
And we've got our trusty guide, Bergey's Manual of Systematic Bacteriology.
A classic. It's like the ultimate field guide for microbiologists, full of flowcharts and tests and all sorts of fascinating details.
Like a detective's handbook, but for bacteria.
Exactly. And today's case involves those tiny cylindrical cells, gram-negative rods.
Ooh, gram-negative. I always get tripped up on that. What does it actually mean again?
Well, you know how bacteria have cell walls, those protective layers?
Right, like there are little fortresses.
Yes. Gram-negative bacteria have an extra layer in their walls, an outer membrane that gram-positive bacteria don't have.
So they're like the double-walled, extra-secure bacteria.
You got it. That outer membrane is pretty important, by the way. It can affect how they interact with their environment and even how they respond to antibiotics.
Interesting. It's amazing how much is going on at the microscopic level.
It really is. So, if we're ready to crack this case, our first clue lies in the oxidase test.
The oxidase test, what does that tell us?
It helps us determine if our bacteria have a particular enzyme called cytochrome C oxidase.
Cytochrome C oxidase, that's a mouthful.
It is, but this enzyme plays a key role in how bacteria breathe, you could say, how they utilize oxygen.
Okay, so it's like figuring out their respiratory system.
In a way, yes. And the results of the oxidase test will send us down different paths on our identification flowchart.
Oh, the flowchart. I love a good flowchart. So let's say our bacteria test positive for oxidase. What's next?
That tells us they have this enzyme and can use it as part of their energy-producing processes. We often find this in bacteria that like to hang out in oxygen-rich environments.
Makes sense. So where on the flowchart does this lead us?
It takes us to a fork in the road. We're now looking at two bacterial families, Vibrio and Aeromonas.
Vibrio. Aren't those the bacteria that can sometimes cause issues with seafood?
You're thinking of Vibrio parahemolyticus, which can indeed cause some pretty unpleasant food poisoning.
Right, right. But there's more to this Vibrio family, isn't there?
Absolutely. The flowchart helps us distinguish between different Vibrio species based on certain characteristics. For example, one of the next tests asks if our bacteria needs sodium to grow.
Sodium, like salt.
Exactly. Vibrio species are a bit picky and require that extra sodium in their diet, so to speak.
So they're the high-maintenance members of the bacterial world.
You could say that. On the other hand, if our bacteria don't need that extra sodium, we're likely dealing with Aeromona.
Okay, I'm following so far. But how do we differentiate between all the different types of Vibrio and Aeromonas?
Well, both Vibrio and Aeromonas can ferment glucose, which means they break it down and produce acid as a byproduct.
Like bacterial fermentation.
You got it.
Oh.
But to tell them apart, we use some other fascinating tests. For example, we can see if they glow in the dark.
Wait, bacteria can glow, like bioluminescent.
Yep. Some Vibrio species, like Vibrio fischeri, are famous for it. They have this amazing symbiotic relationship with certain squids.
I've heard about that. The bacteria live inside the squid and provide light that helps the squid camouflage.
Exactly. And this same bacterium is being researched for its potential in things like pollution detection.
Wow. So much for those tiny, simple organisms, right?
Right. Now, in addition to glow-in-the-dark abilities, we can also look at pigment production, what color they are, and their temperature preferences.
So like some Vibrio like it hot, and some like it cold.
Precisely. Vibriologii, for instance, prefers cooler temperatures. You might find it thriving in the depths of the ocean.
That's incredible. It's like a microbial map of the world, each species adapted to a specific environment. Okay, what about telling Aeromonas apart? What kind of tests are in store for them?
Well, we use a combination of tests to identify different Aeromonas. Some of them might involve checking for luminescence, motility, whether they can move around, and the production of hydrogen sulfide.
Hydrogen sulfide, isn't that the one that smells like rotten eggs?
That's the one. Not the most pleasant aroma, but definitely a helpful clue.
So we're using smell, movement, and even glow-in-the-dark abilities to identify these tiny creatures. Amazing.
It's like being a microbial detective gathering all these different pieces of evidence.
I love it. Now, what about the other side of the flowchart? We started by looking at bacteria that were positive for oxidase. What happens if they test negative?
Ah, yes. If our bacteria don't have that cytochrome C oxidase, it leads us down a different branch, one that focuses on bacteria that can't ferment glucose. And one of the major players in this group is Pseudomonas.
Pseudomonas. I remember those from microbiology class. Weren't they the ones we had to identify based on some colorful pigments?
You're right on the money. Pseudomonas are known for producing some vibrant pigments. Some of them produce fluorescein, a bright yellow pigment that glows under UV light.
Ah, so they can glow too, just in a different way.
Exactly. Different mechanism, different color. Now, some Pseudomonas, like Pseudomonas aeruginosa, produce a different pigment called pyocyanin, which is a beautiful blue color.
So it's like a microscopic rave, all these different bacteria glowing in different colors.
I like that analogy. But these pigments aren't just for show. They can help protect the bacteria from environmental stresses or play a role in nutrient uptake.
Fascinating. So we look for fluorescence, we look for colors. Is that the end of the Pseudomonas identification journey?
Not quite. There's one more step on our flowchart, the nitrate reduction test.
Nitrate reduction. What's that all about?
It tests the bacteria's ability to convert nitrate to nitrate. It's a metabolic process, a way for them to use nitrogen compounds for energy.
Interesting. And not all Pseudomonas can do that.
Exactly. Some, like Pseudomonas putida and Pseudomonas stutzeri, have this capability, while others don't.
Wow. We've gone from a broad category, gram-negative rods, all the way to identifying specific species with unique metabolic capabilities.
All thanks to Bergey's manual and a bit of detective work.
It really highlights how even seemingly simple bacteria can be incredibly diverse, and how important these identification techniques are.
Absolutely. Think about it. These techniques are used in medicine, environmental science, food safety. Understanding how to identify different bacteria is crucial for so many fields.
It really makes you wonder, doesn't it? What other secrets are these microscopic organisms hiding? What other amazing adaptations and relationships are waiting to be discovered?
I don't know, but I'm definitely eager to find out.
Me too. That's the beauty of microbiology. There's always something new to explore. And hey, maybe we've inspired some budding bacterial detectives out there. If you're curious, go grab a copy of Bergey's manual and see what mysteries you can uncover.
I highly recommend it.
Until next time, keep exploring.